Okay, thank you. Let me start today the lecture by a pioneer person in microbiology and a very renowned personality, were still young. The Swarnajit Singh sir told, yes, he is on our platform delivering lecture, means he is young. So today I am very happy to welcome him on behalf of Microbiologist Society, India. And we are recording this lecture on the YouTube. And his ex-student, Dr. Michael, who retired as a professor in microbiology from USA, also joined and going to listen his lecture today. I am really thankful to Dr. Swarnajit Singh for his continuous efforts and dedication toward the microbiology subject. Because of him, such eminent personalities are delivering lecture on our platform. So thank you, sir. Thank you again. And over to Dr. Swarnajit Singh, sir. Please.
I'm very, very happy to have Dr. Joseph Bhagyaraj with us today. He's such a good person. I approached him about a week back and he agreed to give us a talk on microbiology. I'm really very happy that you are here, sir, today. Now, can I request Bhupinder Sharma to welcome?
Okay. First of all, am I audible to you, sir?
Yes.
Yes, okay. Very good evening, one and all who are present here on this platform. On behalf of Microbiologist Society India, as well as MBSI Chandigarh Tri-City, I, Bhupinder Sharma, welcomes you all for the wonderful panel talk on topic soil biodiversity, the present scenario. Before we starting with our actual talk, I would like to welcome or I would like to introduce some eminent personality who are present here on this platform. First of all, I would like to give a very warm welcome to Honorable Dr. Arvind Deshmukh, President MBSI India. Sir is very kind and very sportive. He is the strongest pillar of MBSI and regularly providing such platform to enhance our knowledge regarding recent development in the field of microbiology, biotechnology, and other related scientific fields. Secondly, with a great pleasure, I would like to welcome our eminent scientist, Dr. Savanjit Singh, and more precisely, I would like we all must be very thankful to Dr. Singh for organizing this lecture. Without his dedication, without his support, it is not even possible. So, welcome you, sir, on this platform. And Dr. Samanjit has many achievements to his name. His name is listed in top 2% global scientists for 3 consecutive years. He served in MTCC and Gene Bank, now the first IDA in India. He was awarded with Loyola Environmental Award also. He was the former head environmental biotechnology and microbial biochemistry CSIR MTech. He received Lifetime Achievement Award 2022 by St. Xavier College, Ranchi. His publications are in high impact factor journal and article and are highly cited in the literature and cited around 17,000 times. Sir is also panelist for our today's discussion, and it's an honor for us to have Dr. Singh with us. Thank you, sir, for being here with us. And finally, with a great pleasure, I would like to introduce our keynote speaker, Dr. Joseph Bhagyaraj. Sir is presently Honorable Scientist and Chairman CNBRCD 41 RBI Colony, Anand Nagar, Bangalore. Sir has done his MSc Agriculture, Master Degree in Agriculture and PhD from University of Agricultural Sciences, Bangalore. Sir has completed his postdoctoral degree from New Zealand, Australia, and UK. He is the fellow of Indian National Science Academy, National Academy of Sciences, National Academy of Agricultural Sciences, the National Academy of Biological Sciences, and the Society of Agricultural Biotechnologists. Sir received many awards like SR Vyas Memorial Award, Dr. G. Rangaswamy Memorial Award, Professor T.S. Sadasivan Lecture Award, Dr. V. Agnihotrudu Memorial Lecture Award, Dr. R.Y. Roy Memorial Lecture Award, Dr. Natarajan Memorial Lecture Award, and he also recipient of Distinguished Asian Microbiologist Award 2019 from Asian Mycological Association and several Lifetime Achievements Award. He presented more than 418 research papers. He reviewed around 112 books and chapters. He published 11 books, 14 popular articles, and part of principal investigator, and he's principal investigator of 36 research project in microbial inoculants of sustainable agriculture. He's also part of many. He's also part of many advisory committee like ICAR, DST, DBT, CSIR, ICFRE, UGC, NBA, Rubber Board, Spices Board, IFC Sweden, IACAR Australia, and EU, FAO, UNEP, and GEF. Also, he taught many UG and PG courses in microbiology for the past 50 years, and one of his students, Dr. Michael, who retired from his job but now present here with us. This is also a very honorable thing for us as well. He guided 40 MSc and MPhil students and 23 PhD scholars, currently guiding 2 PhD students. And one of more honorable thing about him, as one of the fungus name has been named after his name, Glomus Bhagyaraji. So, sir, without wasting much time, I would like to hand over the talk to you, and please continue. As thank you for my side.
Thank you very much for the kind words of introduction. And it's always my pleasure. Whenever Dr. Swaranjit Singh contacts me, I can't say no. I have to say yes. So today I thought I should talk on soil biodiversity, the present scenario. I'll be covering soil biota. There will be some animals also coming in addition to microbes. I hope you will tolerate that. Yeah. You know, the term soil means different things to different people. The people living in cities, especially like Mumbai, Delhi, Kolkata, Chennai, Bangalore, then they feel that soil is dust or dirt. When a person, a young boy or a girl goes with a nice pant and a skirt or something, some soil gets into it, they really want to get rid of it because dust has gone into my pant. That is the tendency of the people in big cities. But for a gardener or a farmer, it is the one which gives him the livelihood because he grows crops, he sells it, the whole family lives on that, whatever he gets from that produce. To a civil engineer, let us see, who wants to build a house, and what he does, he just digs the soil so that the foundation can be laid, and he throws it off, so he doesn't need it anymore. But for a biologist, it is a fascinating habitat full of life. So for all biologists, how is soil formed? Soil is formed due to weathering of rocks. And I'll just like to show here, this is the soil profile. Like you see, the top is 0 to 20 centimeters, which has higher level of organic matter, supports the plant growth. And that is A horizon. Below that, the soil between 20 and 100 centimeters is the B horizon of soil, where the deep-rooted plants have their root system spread here. And C horizon is the weathered rock, or the process of weathering is going on there. And the R horizon is the parent rock from where we get the soils formed. It may take hundreds of years for weathering the rock to take place and the soil is formed. And a fertile soil is vital for human survival. Fertile soil is vital for human survival. It is estimated nearly 99% of the world's food comes from crops grown in soil or the livestock maintained on it. And so life, when we cannot see the life below the soil, suppose we just keep our foot and take about 10 centimeters soil, what is said is it harbors millions of biota, but we cannot see them. So life within the soil is hidden. So what is out of sight is usually out of our mind. We don't appreciate it very much. And soil biology or biodiversity is the study of biological systems occurring in soil. And it is alive and supports variety of flora and fauna. Thus, people call it as a factory of life. In fact, United Nations declared 2010 as the International Year of Biodiversity. And we also know that the years from 2011 to 2020 is a decade of biodiversity. And so soil biologists, they study the organisms living in soil. And another important thing is, if we estimate the total biodiversity on Earth, 25% of them come from soil. 25% of the Earth biodiversity is from soil. And how can we classify the organisms present in soil? So it is classified based on the body size. If it is small, 1 to 100 micrometers, which includes bacteria, fungi, we call it as microflora. And little bigger, 5 to 120 micrometers, which includes protozoa, archaeozoa, we call it microfauna. Still bigger, 80 micrometers to 2 millimeters, which includes columbolas, nematodes, we call them as mesofauna. And still bigger, 500 micrometers to 50 millimeters, which includes earthworms, termites, etc., we call them as macrofauna. The inventories on soil organisms is limited compared to the above ground biota. For example, vascular plants. A lot of studies have been done on vascular plants, but when we think of soil organisms biota, much less studies have been carried out. Again, if the organism is big, then more studies have been done in soil. If the organism in soil is small, lesser amount of studies have been made. And that is what is given here. In case of vascular plants, the total estimated species is on 300,000. But how much we have studied so far? 270,000. That means as biologists, we know 90% of plants. When we come to ants, we know only 58%. When we come to columbola, we know only 27%. When we come to protozoa, we know only 7%. But when we come to microbes like bacteria and fungi, we know only 3% of the organism existing in nature. So what soil biota contains macrofauna, mesofauna, microfauna, algae, fungi, bacteria, ones which are smaller than that, like viruses, viroids, prions, and so on. We will just briefly study each one of these groups, starting from the bigger one, the macrofauna. Macrofauna includes earthworms, mollusks, insects, millipedes, etc. So what is the major role played in soil is the organic matter breakdown and decomposition. They also improve soil structure, aeration, water infiltration, and so on. They also predate on soil microorganisms, thereby they maintain the biological equilibrium. And I have just included some photos of macrofauna like thrips, termites, ants, centipedes, millipedes, earthworms, and so on. And also includes variety of beetles, also spiders, slugs, snails, and so on. This is a very interesting photograph I could get. You can see the size of the earthworm, the length of the earthworm. It is as tall as the man himself. It is a giant earthworm, Rhinodrilus preoli, which has been found in Brazilian Amazon forests. So it's like this. We also have another earthworm in Western Ghats, and this is called Ravida species. One can even mistake this to be a small snake, as big as a small snake. So these are all the macrofauna. Coming to the second category, mesofauna, which includes springtails, rotifers, nematodes. More studies have been done on nematodes, and we know that nematodes, some of them are plant pathogens and some of them are non-pathogenic, but they regulate the microbial population in soil. And I've just included some of the photographs of mesofauna, tardigrades, nematodes, rotifers, mites, and so on. Coming to the third category, microfauna, which includes smaller organisms like protozoa and archozoa. The main difference between protozoa and archozoa is archozoa do not have mitochondria. That's the only difference. And both these groups, they are important predators of bacteria and again regulating the population in soil. And some of them are pathogenic and they are found in drinking water. In fact, in and around Bangalore, we have this problem now. There is contamination of Giardia species, which causes giardiasis, which results in foul-smelling, watery diarrhea in human beings. I just included a few photographs of amoeba, flagella, and ciliate. Coming to the next category, algae. We know that algae are all photosynthetic organisms. They prefer to live in marshy environment. And they also form symbiotic association with certain fungi, and they are called as lichens. And so the algae and lichens contribute to soil formation, soil aggregation by adding organic matter to the soil. And some of them also fix atmospheric nitrogen. We know blue-green algae, Lake Nosta, Canabina, which fix atmospheric nitrogen. Coming to the next group of biota, fungi. So it could be unicellular yeast, or it could be bigger ones like mushrooms and puffballs. And what is the role played by these fungi in soil? Decomposition of organic matter, soil aggregation, nutrient cycling, and biocontrol of plant pathogenic organisms. And some of them are edible also. Mushrooms, we know, truffles are all edible. And some of them cause diseases of plants, animals, and human beings. So I've just included yeast, penicillium, mycorrhizal fungus, ectomycorrhizal fungus, mushroom, and a nematode trapping fungus. And coming to the next category, bacteria. And here we have archaea and bacteria. And now bacteria includes actinomycetes, called as actinobacteria. And we know that archaea are very common in extreme environments, very hot or very cold environments. And bacteria is more common in soil environment. And we have, when we talk of the soil bacteria, we recognize two categories, autochthonous bacteria and zymogenous bacteria. Autochthonous bacteria are those which are indigenous, present always. When we take a soil sample and look for the bacteria, they are present now. After 6 months, you take again, they are the same bacteria are present. Whereas the second category, zymogenous bacteria, they are found when a particular substrate is present. For example, suppose some cellulose is added to soil, when the cellulolytic bacteria develop there. And they are present till the cellulose is degraded. Afterwards, they disappear. So such organisms are referred to as zymogenous bacteria. What is the role played by these bacteria in soil? In nutrient cycle like nitrogen cycle, phosphorus cycle, and so on, and organic matter decomposition, production of industrially important secondary metabolites like enzymes, vitamins, and so on. Some of them cause diseases in plants and animals. And some of them cause diseases in plants and plants, we said. And one of them, which is very important now, is the bacterial blight of pomegranate. India is not able to export pomegranate because of this bacterium, Xanthomonas axinopodis. And we also know they cause animal diseases like anthrax and human diseases like typhoid, cholera, dysentery, and so on. And we also have organisms which are smaller than bacteria, the viruses, which have the central nucleic acid, RNA or DNA, surrounded by a protein coat, and they cause diseases in plants, animals, and humans. And we also have mycoplasmas, which is bacteria without cell wall, again causing diseases in plants. And we also have viroids, which is have only the central nucleic acid, but no protein coat. And opposite to that is the prions, which is only the protein coat and no nucleic acid. And again, these also are being reported to form diseases in plants. And here, some scientists have studied whether there is any correlation between the size of the organism and the biomass it contributes in the soil. So, and some other group of scientists have also studied whether there is any correlation between the population density and importance to biological activity in soil. For example, suppose we keep our foot and take about some soil up to a depth of 10 centimeters, there could be billion protozoa, but there could be only two earthworms. So what we say is the two earthworms are biologically more important than the billion protozoa. So that is, there is no correlation between population density and the importance to the biological activity. Taking the size and biomass, let me take this. And we know the microbes are very small in size, bacteria and fungi, but they contribute to 80% of the soil biomass. Macrofauna, which are big in size like earthworms, potworms, they contribute only 14% of the biomass, soil biomass. So there is no correlation. And yes, the essential ecosystem services provided by the soil biota has been studied in detail by many workers around the globe. And what are the various ecosystem services they provide? Decomposition and cycling of organic matter, regulation of nutrient availability, suppression of pests and diseases, and maintenance of soil structure, and gas exchange and carbon sequestration, soil detoxification when a xenotoxic material like pesticide is added, they undergo decomposition and slowly they are detoxified. Many of them contribute to the plant growth like root nodule, rhizobia, mycorrhizal fungi, plant growth promoting rhizome microorganisms, and so on. Some of them help in the pollination of horticultural crops. And here, especially a soil nesting insect called a solitary bees play a very important role in pollination of horticultural crops. So these are all the various services provided by the soil biota. We can see here the soil, various services provided by the soil biota, and we can say that soil structure and maintenance, carbon cycling, decomposition of organic matter, climate regulation. When I say biotechnology, biotechnologists look to the soil for organisms which produce bioactive compounds like enzymes, antibiotics, and so on, and plant growth production, and also nutrient cycling, and biological population regulation is very important in soil. So these are all the various services provided by the soil biota. And Millennium Ecosystem Assessment defines ecosystem services or the benefits people obtain from the ecosystem. That is the definition. And what are this? These services have been categorized into 4 groups. What are they? They are supporting services. Example like nutrient cycling, soil formation, etc. Second category is provisioning services. That means they provide us food, fuel, fresh water, and so on. The third one is regulatory services like wastewater, waste purification, climate regulation, etc. Cultural services like recreation aesthetics, nice parks where we go out for recreation. And soil organisms provide all these services like nutrient cycling, control of pests and diseases, degradation of waste, harmful chemicals, and production of food and fuel. So the soil organisms provide all these ecosystem services. And just take a couple of these ecosystem services provided by soil biota and then see how this takes place in soil. And we know that the plants and animals are all buried in soil. And when they reach the soil, and these plants, for example, they are rich in sugars, cellulose, pectins, proteins, fats, phenols, alkaloids, etc. And they are acted upon by soil biota. And how are they acted by soil organisms? First of all, they are acted out by macrofauna. So when the mollusks, millipedes, insects attack the litter when it is buried in soil, and when we take a leaf, for example, they puncture the leaf epidermis. This process is called fenestration. I will show you a photograph a little later, and increases the surface area for the microbial attack. And so, and another very interesting experiment have been done, very simple but interesting. What they have done is they took the bags with holes of different sizes, a bag with holes of different sizes. For example, here in this 7 millimeter mesh hole, that is the size. In another bag, they had a very smaller hole, 0.5 millimeter hole. And what they did, they added lot of leaf and buried it in the soil.
बहुत सारे रख दिए हैं 4
कहाँ रखते हैं
कार में रखे पीछे
कार में रखे ना मेरे को सुबह थोड़ी मिलेंगे बाहर निकाल
Sorry to interrupt you, sir.
Okay.
Professor Sivesh Singh, please mute yourself.
Yeah, please.
Okay, thank you.
So I was telling about the 2 bags, right, with different hole size. So one bag was having a bigger hole, 7 millimeter hole. Other bag was having only 0.5 millimeter holes, very small holes. They added lot of leaf matter in this, buried it in the soil, and after 4 months they took it out and then studied the extent of decomposition. Where the size was, the hole was 7 millimeters, where the bigger holes, there was 95% decomposition, whereas where the hole size was very small. There was only 35 millimeters. That is mainly because the bigger hole allowed the microfauna, flora to enter and decompose. Whereas the smaller size, prevented the environment entry of the bigger invertebrates. So there was no less decomposition. And this is what the defenestration, you can say, how the animals act on this and then make it available, make the surface area more for microbial decomposition. And and when we say that, when an organic matter is added on to the soil, especially the plant material, the mollusks, wood-lice, millipedes, earthworms, they macerate and macerate the litter and pulverize it. And fauna like earthworms, insects, they transport it to the deeper layer of the soil. Again, earthworms and potworms further macerate it and mix it in the soil so that we have the organic matter completely pulverized and we have the compost that is added on to the soil. And even with this, all this reaction, there will be some part of the organic matter which will not be fully decomposed. And they are very resistant for decomposition. And they undergo very, very, very slow decomposition. They are also black in color and they are referred to as humus. And they undergo very slow decomposition and make the nutrients available for plant growth. And this is a very, very interesting experiment done by collaboration between microbiologists and a zoologist. Okay, so what we have done, we have taken a Pseudomonas fluorescens, which is a plant growth promoting rhizobacteria, and they have taken 3 different species of earthworms. Okay, so these were the 4 treatments. One is the control, where no earthworm was added, and under the second treatment, they added this earthworm, third treatment, this earthworm, and fourth treatment, this earthworm. And what they examined, to what depth this PGPR is taken down by the earthworms. So in case of control, no earthworm was added. Because of the water movement, it went up to 10 centimeters deep. With this earthworm, it went up to 20 centimeters depth. And with these two, it went up to 40 centimeters. Again, this particular earthworm has taken more population of rhizobacteria. So that means these helpful soil bacteria are taken to a deeper depth by these earthworms, thereby the deeper plants with deeper root system are benefited by these beneficial bacteria. Very simple, but giving lot of information. Second important ecosystem service provided by soil organisms are the symbiotic nitrogen fixation. And we have symbiotic, asymbiotic, and free-living nitrogen fixation. And we know nitrogen is one of the major plant nutrient, and it is present in 80% in the atmosphere, and plants cannot use it directly. But there are some bacteria which can fix atmospheric nitrogen, and all of us know about the root nodules in leguminous plants, and it makes it available for plant growth. And we know that rhizobia forms the root nodules in leguminous plants. And again, in addition to that, we have another organism which can form root nodule as well as the stem nodule, Azorhizobium colinodens, and this forms root as well as stem nodules. And we also have the anactinobacteria like Frankia, which forms nodules in Casuarina. With the inoculation with these organisms, it has been shown that 50% of nitrogen fertilizer application can be reduced. I've just introduced one slide, so containing a root system of a legume, showing the root nodules. More the pink in color, that means it's a better nitrogen fixer because it has a concentration of leg hemoglobin is more. When the concentration of leg hemoglobin is more, it's an indication that it is a better nitrogen fixer. And I just introduced one photograph of stem nodule formed in a green manure plant, and it's caused by Azorhizobium colinodens. And the plant that is commonly used is Sesbania rostrata, very, very commonly used in Tamil Nadu. The farmers grow this before raising the rice crop, puddle the thing after about 6 weeks, and then transplant rice. And this is Casuarina, showing the root nodules. So you can see this is caused by Frankia and Actinobacteria. The nodules are quite big. It could be as big as the size of a table tennis ball. And this is you can see the difference between inoculated soybean and uninoculated soybean with rhizobium. And a lot of studies have been done in our country with the rhizobial inoculation in various crop plants. In case of chickpea, and up to 20% increase has been reported. In case of pigeon pea, up to 30% increase in yield has been reported. Coming to the second category of nitrogen fixation, associative nitrogen fixing bacteria, they do not form nodules, but they enter the epidermal cells and the outermost cortical cells, sit there and fix atmospheric nitrogen. Classical example is Azorhizobium, and another example is Gluconoacetobacter diazotrophicus, which is a sugar-loving bacterium, and as the name itself indicates, they are found in plants like sugarcane, sweet potato, sweet sorghum, and so on. The third category is the free-living nitrogen-fixing organisms like Azotobacter. They do not enter the plant cells. They colonize the rhizosphere and fix atmospheric nitrogen and make it available for plant growth. Same thing applies to cyanobacteria, earlier called as blue-green algae, and they also fix atmospheric nitrogen. I've just included a couple of slides to show the increase in yield due to Azospirillum inoculation. In Amaranthus, it is 36% increase has been reported. In the free-living nitrogen-fixing bacterium Azotobacter, like in cabbage, 19%, in tomato, 28% increase has been reported. In cyanobacteria from Madhya Pradesh, 17% increase in yield of rice has been reported. And we know in cyanobacteria, the heterocysts are the site of nitrogen fixation. And this is a photograph which I took in Tamil Nadu in Kanyakumari district, where the farmer is mass multiplying the cyanobacteria himself. So what is he made a trench and put a polythene sheet and added some water and sprinkled some soil and then got inoculated with the cyanobacteria, which he got it from a laboratory. And in 3 weeks, it multiplies so fast. And this he can either dry and use later, or when it is fresh, he inoculates into the rice field before transplanting the rice seedlings. And just a nitrogen cycle. The nitrogen is present in the atmosphere, and it is fixed by the nitrogen-fixing organisms, and it is fixed in the form of ammonical form, which most plants do not take ammonical form of nitrogen. They prefer to take nitrate form of nitrogen. Again, there are bacteria in soil like Nitrosomonas and Nitrobacter, which converts ammonical form into nitrate form, which is taken by the plants. And there are again some bacteria, denitrifying bacteria, which act on nitrates present in soil and then release the nitrogen, which goes back to the cycle. So all of us know about this nitrogen cycle. Coming to the next major plant nutrient, that is the phosphorus. And most of the tropical soils are deficient in phosphorus, and which our country, most of the soils are deficient in phosphorus. And in addition to that, when a farmer adds a phosphatic fertilizer to the soil, 75 to 80% of it is converted to unavailable form, which plants cannot take. In acidic soils, it is converted to iron and aluminum phosphate. In sodic soils, alkaline soils, it is converted to calcium phosphate, which plants cannot take. And but fortunately, we have many phosphate solubilizing bacteria in soil. For example, bacteria like Bacillus polymixa, Pseudomonas striata, fungi like Aspergillus saumori, Penicillium digitatum, which can act on this unavailable form of phosphorus through the production of organic acids and make the available form of phosphorus for plant growth. And plants prefer to take phosphorus in the form of H2PO4 ion. So these bacteria convert and release this form, and which the plants can take. And there are cheaper sources of rock phosphate also available in our country. And farmers have tried using rock phosphate along with phosphate solubilizing microorganisms and use it. And one can screen in the laboratory the ability of the phosphate solubilizing bacteria. You can see the bigger the zone of solubilization, it's a better P solubilizer. I've just included one phosphate solubilizing bacterium, one fungus, which is commonly mass produced. And lot of studies again have been made in our country on phosphate solubilizing microorganisms. And in cabbage, 56% increase has been reported, in tomato, 37% increase. Another group of organisms which help in phosphorus utilization is the mycorrhizal fungi, arbuscular mycorrhizal fungi. And these are the 4 genera which are very commonly present throughout the world, including India. And of these 4, the one which is most common is globus. They help in the uptake of phosphorus. And these mycorrhizal fungi, they colonize 90% of vascular plants. 90% of vascular plants are colonized by these mycorrhizal fungi. Very few families are not colonized by mycorrhizal fungi. Plants belonging to Brassicaceae, Cyperaceae, Polygonaceae, Myrtaceae. These are few exceptions which are not colonized, whereas all the other plants are colonized by these fungi. I've just included one slide just to show the effect of inoculation with mycorrhizal fungus. This is okay. And here, in addition to phosphorus nutrition, they also have other beneficial effect on plant growth. They have synergistic interaction with other organisms, beneficial organisms like nitrogen fixers, PGPRs. They also protect the plants against plant pathogens, and they also help in the plants to overcome drought tolerance. They also produce growth promoting substances like indolactic acid, cytokinin, and its overall effect, they help plant growth. Coming to the next category of microbes, plant growth promoting rhizomicroorganisms, and they increase plant growth through the production of phytohormones like indolactic acid, gibberellin, cytokinins, and they also suppress the plant disease. So these are the 2 mechanisms by which they help plant growth. And we have a variety of organisms which has been studied globally. The one organism is Pseudomonas fluorescens. A lot of studies have been made, but there are some organisms where very little study has been made, like Methylobacterium, Azoarchus, Pantia, Penibacillus. So very less studies have been made, especially in our country, on these PGPR. So there was a scientist, she worked in my laboratory as a DST women scientist, and she worked on this PGPR, newer PGPRs, and she worked on French bean, and she developed the best PGPR for inoculating French bean. That happened to be Pantia agglomerans. She did the glasshouse studies, then took it to the field studies, followed by the farmers' field studies. And now this is a practice commonly followed in and around Bangalore by the farmers. And coming to another group of organisms are the biocontrol agents. And we know that various crop plants here I have listed, and the various diseases on them, serious diseases, and which is the biocontrol agent. I will take one example, pigeon pea. Wilt caused by Fusarium is a very common disease, and the biocontrol agent is Trichoderma viride. And so if you see the last column, the biocontrol agent, you can see Trichoderma is the most predominates, and it's the most common biocontrol agents which can be used for biocontrol of plant diseases. And coming to insect pathogens, again, all of us know about Bacillus thuringiensis, and which is very effective in controlling the bollworm of cotton. And coming to pesticide degradation, again, degradation of pesticides is done by microbes, and some of the pesticides are used as carbon and energy source by these organisms, and thereby making it to disappear from soil. And extension to other fields, as I told earlier, the biotechnologists look into the soil as these organisms present in soil act as a source of antibiotics, vitamins, enzymes, and so on. I've just given some examples here. For example, gibberellic acid is industrially produced using the fungus Gibberella fusicore, and the proteolytic enzymes are produced by Martirella species. And about the antibiotics, all of us know about streptomycin, penicillin, and so on. and proposed economic value of the ecosystem service. Here, what can we give some amount, the economic value for the ecosystem service done by or given by these soil biota? Unfortunately, till today, we do not have a report from India which needs a collaboration with the microbiologist or soil biologist with economists. But attempt has been done in US and that given a value. For example, nitrogen fixation, they have given a value $90 billion per year. Biocontrol of pests, $160 billion per year. So I think this type of work has to be done in our country. And coming to what are all the, sorry. Yeah. What are the various threats to soil biota? How the soil organisms are threatened or they are disappearing in some cases. And the main cause is the pollution. When we say pollution, industrialization, and who is behind this industrialization? This is the human beings, we. And again, other things are modern agricultural practices. And instead of having mixed cropping, we go for monocropping. And climate change is another reason. Overgrazing by animals, loss of above ground biodiversity, introduction of alien species, soil erosion, land degradation, and forest fires. All these are responsible for the threats to soil biota. And the same thing is presented in another way. The potential threats are the human intensive exploitation. Okay, can we conserve soil biodiversity? A lot of importance is given now to conservation of soil biodiversity. So what are the areas that are suggested to conserve soil biodiversity? Is diversification of the crops, soil erosion control, fire management, agroforestry, bioremediation using biological materials, soil amendments, no tillage, I would make it as less tillage. And so these are the things which can conserve soil biodiversity. And this is a very nice slide. Children love to play in soil. I have also, as a small boy, I have also played with soil. And how many of us know what the biodiversity in soil? Here is an interesting picture. A primary school teacher asked the children to write what they know about the life in soil. This is the drawing made by one of the child. So you can see the picture. You can make out the soil. The child knows there are certain living things in soil. And another teacher wanted to know how much the children know about soil. When she talked to the 4 to 7 year old children, asked them what do they know about soil. One child said, without soil, there is only a big hole. Another child said, trees grow on soil. Another child said, my ball won't bounce back. Another child said, without soil, we will tumble down. Going to the little older children, 7 to 9 years, one child said, plants grow in soil. Correct. In soil, there is seed. I don't know, must have gone below a tree and must have seen a lot of seeds. And another child said, foxes live in caves. And so this is what another child said. And still older children, 9 to 11-year-old child, they said, one child said, trees grow in soil. And it went further. They produce oxygen for human and animals. That means the child knows the importance. And another child said, in soil, I can find earthworms that I need to catch fishes. So the children know the importance of soil. Are you? Okay. And we come United Nations declared, as I told earlier, 2010 as the International Year of Biodiversity, and lot of importance was spotlight was given to the biodiversity in soil. And in 2010, they also brought out the European Atlas of Soil Biodiversity. It was brought out by the European Commission. And the International Technical Workshop was organized by FAO in 2012. And the title of the workshop was Managing Living Soils, and it was held at December at Rome. And what came out of this workshop is a program called Global Soil Partnership Program. What does this program actually emphasizes? It emphasizes the management of soils for food security, agriculture, mitigation of climate change, poverty alleviation, and sustainable development. And this Global Soil Partnership Program has 5 pillars of action. And 1 is soil management, 2 is investment, 3 is research, 4 is information and data, and 5 is standards. So pillar 1 deals with managing living soils, and pillar 3 is research. These are the 2 important components of this program, which continues even now. And again, United Nations, sorry. Yeah. Oh. And we know that United Nations declared 2015 as the International Year of Soil Biodiversity, and also 5th of every year as the World Soil Day. And in 2013, I told you earlier, the European Soil Biodiversity Atlas was brought. But in 2013, it was decided by FAO to bring out the Global Atlas on Soil Biodiversity. Nearly 100 scientists from 26 countries were invited to contribute to this atlas, and in their area of specialization. For example, if 3 scientists were recommended to write on nitrogen fixing bacteria, 3 on earthworm, something like that, 4 specialists. And they also said it is for common man. So it should be in a very simple language, but should have beautiful photographs. So it was, I just have introduced here, this is the first page of the Global Soil Biodiversity Atlas, and this is the last page. And okay, before conclusion, I would like to say what is happening at present. And here, what the work that has been done very clearly shows that children playing with soil and gardeners who are working with soil, they come across the bacterium Mycobacterium vaccae, which enters either through cuts or wounds in the body, or they inhale the bacterium. This bacterium is known to produce a happy hormone called serotonin. Okay, so that makes them feel very happy, decreases their anxiety, make them feel very relaxed, and it also improves the various mental activities most closely associated with learning and problem solving. And coming to the another work, in the National Institute for Health and Welfare, Finland, studied the incidence of asthma in rural and urban children up to the 6 years age. What they found, incidence of asthma was 4% in rural children exposed to soil with beneficial microbes, and it was 29% in urban children exposed to traffic pollution. So this was confirmed later by studies done in other parts of Europe and USA. And the conclusion they could draw from this, early life exposure to soil or dirt matters a lot, increases the immunity in the children. So I think we should not prevent in future, at least preventing our children playing with soil and lost. And coming to the conclusion, Soil is a reservoir of several organisms. Soil biologists study them in isolation. The person working with protozoa never interacts with the person working with soil bacteria and vice versa. And soil biodiversity is critical for plant growth, climate regulation, ecosystem functioning, disease control, and human health. There is need for collaboration among soil biologists working on different groups of soil organisms. Coordinated studies are also needed between soil biologists, soil chemists, agronomists, biochemists. You can extend this specialization to any extent. More research is needed to understand the complicated and interlocked processes carried out by organisms in soil in order to derive from them benefits for mankind. If the soils are healthy, the plants growing on them are healthy. And the animals which are feeding on those plants are healthy. And the man feeding on those plants and animals will be very healthy. Thank you very much. I'll be happy to answer any questions. They can put it in the chat box also.
Thank you, Dr. Joseph Bhagyaraj. It was an excellent, wonderful talk. And we learned so many things from your talk. And we are really enlightened about fungi and plants. Does anybody have a question you can ask? Anybody has a question? Okay, if not,
Dr.
then
Bhagyaraj, I have one question. This is Michael.
yes,
So,
yes, Michael.
thank you for excellent talk. So I was very interested in the Mycobacterium vaccae that produces serotonin. This one.
Mycobacterium vaccae, which produces serotonin, which is a happy hormone. So it makes the people very happy. That gives.
Yeah, I've never heard of this before. So this is very interesting.
Very interesting, really. Dr. Joseph, I'll ask you one or two questions.
Yes, sir.
Which we don't know actually. How can I get a global soil biodiversity atlas? I want to get one.
Okay. This one, you can go to the European website, European Commission website. I made it ready here. Global Soil Biodiversity Atlas.
Yes.
You can go that, and you can have this particular slide. So you just go to this HTTPS, what I have given. First, Global Soil Biodiversity Atlas, and then come to the here. And this soft copy you can download freely.
Okay.
It is free. But if you want a hard copy for your library, it costs 25 euros.
Okay.
But one can download freely the soft copy.
I think downloading freely is better because they keep on updating.
No. Not in this case. In fact, earlier it was 25 euros. I was told that recently they brought it to 23 euros.
Okay. That's right. Okay. And but I think the soft copy will be better because they regularly update it.
Yes.
Yeah. So I'll definitely get one copy. Okay. Do all vascular plants have AM fungi?
Not all of them actually.
Okay.
About 90% of them have the colonization by mycorrhizal fungi. There are some plants belonging to families like Brassicaceae, the same as Cruciferae, Myrtaceae, Polygonaceae, and they do not form the mycorrhizal association. People have investigated why they do not form the colonization in these plants. They found that the concentration of isothiocyanate is higher in the roots of these plants, and which inhibits the colonization by mycorrhizal fungi. That is what has been reported.
Okay, that's very interesting, really. And are the people maintaining consortia of the microbes for use?
Of
Mixed culture, say many cultures together, consortium.
consortium, I mean, different organisms, or are you talking
Yes.
of mycorrhiza?
Different organisms.
Sir, you are maintaining the UTech is maintaining.
No, we are maintaining pure cultures.
Pure
But now,
cultures, okay.
yeah,
But you
yeah.
can mix them and use.
But now people are conserving the consortium.
Consortium.
Yes.
I think I don't see people doing it. Neither in NBAM they are doing it.
Okay.
Not as a consortium, but they want a organism to supply nitrogen, phosphorus, and potassium. They
Right.
take the three organisms, mix it, and then use it.
Right.
And then that's a
Actually, the consortium together work better than one culture.
Correct.
Yeah, that's why the concept is growing now to preserve the consortium.
Yeah.
Very nice. Very nice. And I think there is one question in the chat box. Why India is not working on this field?
Pardon? What question?
Why India is not working in this field?
India is working, actually. Soil biodiversity, many people are working.
I see.
In fact, I forgot to tell in this things that for this global soil biodiversity atlas, I was telling that they were inviting people, for example, nitrogen fixing bacteria, 3 scientists from different parts of the world to write and then contribute to the global biodiversity atlas.
Right.
So for mycorrhizal fungi, 3 of us, one from US, me, and one from Brazil, were invited to write on mycorrhizal fungi. So it so happened I was the only scientist from India to contribute to the global soil biodiversity atlas. So
that's good. So if there is no more question, does anybody have a question?
Hello,
Yes.
Dr. Baljeet Saran from Haryana Agricultural University, sir.
Yeah.
Yes.
And thank you very much, sir, for very nice and very informative information for soil scientist and microbiologist. Sir, this global soil biodiversity atlas, it is having the information based on cultivable microbes only, or on the basis of metagenomic approach?
I don't think it has some metagenomic approach. It's only on cultivated organisms.
So, sir, only cultivable, they are 2 to 3% microorganisms.
Yeah, that's true. But this was produced in 2016. At that time, it only on cultivable. Yeah.
Okay, sir.
Okay.
Good. Thank you. Thank you, sir.
Thank you.
I think there is one more question in chat box by Madam Aparna George. Let me present to sir. Sir, she want to ask that how long does it take for nodule formation in agroforestry legume trees?
Yeah, even in the seedlings, they do form nodules. They need not have to become trees to form nodules. Even the seedling form nodules.
Okay.
Because we have, I myself have worked on Lucina leucocephala extensively. In the seedling stage itself, it forms the nodules. Yeah.
Okay, if there is no more question, then Bhupinder, you can please give the word of thanks.
Okay, thank you, Sanjit sir. Okay, first of all, those who joined a bit late, I would like to tell them that before the actual start of the session, Dr. Bhagyaraj said that he is 82 years old. But sir, I personally don't feel it because your energy level is unmatchable. You are inspiring many young lads like me. And age is just a number. If a person is dedicated towards his or her work, I feel that age is just a number if someone is dedicated towards his or her work. So thank you, sir, for sharing your vast experience with us and giving your precious time. This lecture is very informative and very amazing. One line I would like to say that बचपन में हम एक पोएम पढ़ते थे कि जिसकी रज में I hope many of हमने पढ़ी हो कि जिसकी रज में लोट लोट के बड़े हुए हैं, घुटनों के बल सरक सरक के खड़े हुए हैं. रज means धूल, टन, मिट्टी. So soil is the part of our culture as well. So soil biodiversity is part of our social custom as well, कि बच्चों का बचपन मिट्टी के साथ ही है. Without soil ये पॉसिबल ही नहीं है कि बच्चों ने बचपन जिया है. तो सर, all soil microbiologists and all those person who is working with soil, वो एक्चुअली हमारा बचपन भी प्रिजर्व कर रहे हैं और हमारे फ्यूचर जनरेशन के बचपन को भी प्रिजर्व कर रहे हैं. So we are highly thankful to you guys. And finally, we all must be very thankful to the man who is behind this talk, Dr. Savanjit Singh sir. Sir, your sincere dedication must be appreciated. You are doing lot of hard work to arrange such a talk on this platform by communicating with eminent scientists from various fields and also insist them to deliver lecture on our platform. Thank you, sir. We are very lucky to have you with us.
Thank you very much.
So-
Thank you. Thank you.
And finally, I would like to thank our audience as well, because they listen this informative talk very patiently. Thank you, audience, and thank you everyone for joining us.
And
So
thank
that's
you
all
very
from my
much.
side.
Oh, it's a
Thank
pleasure.
you very much, Bhupinder, for conducting the talk so well. Thank you so much.
Thank you. I'm so
Thank you,
happy to see.
Dr.
Welcome,
Joseph.
sir. Welcome.
I'm so happy to see Dr. Krishna Raj also there.
Yes,
I'm so happy to see Dr. Michael. So
Yes.
it's a
Yes.
pleasure.
Thank you. Thank you.
and thank you, Dr. Joseph, once more for the wonderful talk you gave. Thank you so much.
Thank you very much.